Hello, I am Shweli Dash. Welcome to the Knowledge Club Online. I discussed in our previous lecture about the three stages of the viral replication, that are the attachment, penetration and uncoating part. I also discussed about the DNA genome biosynthesis process. If you missed that video, please check our YouTube channel or click at the I button above this video. Today, I will discuss about the biosynthesis part of the RNA virus genome and also discuss about the rest part of the viral replication, that are assembly and the release part. Then tackle. At first, if you are new knowledge gainer, don't forget to subscribe our channel and get our update at the bell button. So first I am starting with the biosynthesis process of the RNA virus genome. Replication and transcription of RNA viruses are similar processes because the viral genomes are usually either an mRNA, that is plus strand RNA, or a template or mRNA, that is minus strand RNA. At the time of replication and transcription, a double-stranded RNA intermediate is produced. Most RNA viruses replicate in the cytoplasm of host cell. This is the main difference between a DNA virus and RNA virus. If you rememorize the previous video where we learned about DNA virus replication, which replicate in the nucleus of host cell, the RNA structure determines the mechanism of mRNA generation and processing of proteins, which are plus RNA viruses and the minus RNA viruses, means the negative RNA viruses. So first I will discuss on the positive RNA viruses. This comprises a large group of viruses including picorma, toga, flavi, cassi and coronaviruses. The positive RNA gene resembles mRNA. In early period, following entry of the virus into the cells, the viral DNA binds to ribosome, acts as a mRNA, and is translated in its entity into one large polypeptide. The large polypeptide is then cleaved by proteolysis to give as products RNA polymerase, protease enzymes, and new capsid proteins. In the late period, the new RNA polymerase copies the positive RNA strands into negative RNA strands that are used as template for replication, leading to synthesis of large numbers of new positive RNA strands. The newly synthesized positive RNA is packed in the progeny viruses and used to produce more amount of structural proteins. Now come to the last point of the viral genome biosynthesis, that is the negative strand RNA virus genome. In this group, there are orthomixo, paramixo, rhabdo, philo and bina viruses. The normal cellular enzymes are unable to replicate RNA as the cell has no RNA-dependent RNA polymerases. So, the virus has to pack its own polymerases. The negative stranded RNA genome is not infectious by itself, and a polymerase must be carried into the cell with the genome, which is associated with the genome of the virus as part of the nucleocapsid. To make and transcribe positive RNA strands, that is equivalent to mRNA. In early period, that means the early stages of replication are concerned with the synthesis and transcription of positive RNA strands, which is done by utilizing mRNA as a template. Both short and full-length transcripts are produced. In the latter period, the short positive RNA strands are translated into several viral proteins and enzymes which are required to form the new virions, while the full-length positive RNA strands act as templates for progeny, that is the negative strand RNA. With the accumulation of negative RNA strands, they also serve as templates for additional short positive RNA strands that are necessary for synthesizing structural proteins and polymerases to be packed into the the progeny particles. This case is the exception for influenza viruses. All negative strand RNA viruses replicate in the cytoplasm, but the influenza virus replicates in the nucleus. So, these are the overall structure of biosynthesis of various virus genome. Then proceed towards the next step of the viral replication cycle. The fifth stage is the assembly. The assembly stage is also called the morphogenesis. Following synthesis of viral proteins and viral nucleic acid, there is a stage of assembly which is known as morphogenesis, and that makes the end of the eclipse phase. The incorporation of the nucleic acid into a nucleocapsid is facilitated by the formation of protein and nucleic acid in close proximity and by packing signal. Which are the specific sequence on viral nucleic acid that determine interactions between core and internal capsid proteins. The site and mechanism of assembly of virus particles in the cell depend on where genome replication takes place and whether the final structure is a naked capsid or an enveloped virus. Here, assembly may occur in nucleus or cytoplasm. In nucleus, DNA viruses except pox virus are assembled in the nucleus and requires transport of the virion protein from the cytoplasm into the nucleus. These proteins must have the appropriate peptide signals for nuclear transport in addition to structural features. In the case of cytoplasm, assembly of RNA viruses and pox virus occurs in the cytoplasm. Here, another main part is the outer covering. In case of naked viruses, they can complete their maturation in the cytoplasm or in the nucleus. Infective viruses are present in the cytoplasm for a brief period of time before they are released when the cell bursts. But in the case of enveloped viruses, they complete their maturation at the time of budding. The site of budding is determined by the type of viral genome and the protein sequence of glycoproteins. After the envelope proteins are formed during the stage of synthesis, they are glycosylated or meristylated, then transported and inserted into a cellular membrane. Thus, herpes virus acquires their envelope by budding through nuclear membrane. The flaviviruses, coronaviruses and pioneerviruses acquire their envelope by budding into the endoplasmic reticulum and Golgi membranes, and may remain in these organelles. Most other enveloped viruses bud through the plasma membrane. The enveloped glycoproteins are annexed in patches cross-linked to an inner viral coded protein layer that is equivalent to the outer capsid. The viral nucleocapsids are attached to these modified patches of cell membrane and pinch off pieces of the lipid bilayers membrane, which brings to an end of the assembly process by completing the formation of the envelope. Errors are made during viral assembly in this case. The assembly stage. Empty viruses and viruses containing defective genomes are produced. The particles to infectious viruses are also called particle to plaque forming ratio is usually greater than 10 and attains 10 to the power 4 during rapid viral replication. Defective virus can interfere with and prevent virus production. Defective interfering particles. The last stage of the replication cycle is the release. Release of the newly formed virus particle. The assembled viruses are released to the extracellular fluid by one of the two basic mechanisms, that is lysis or budding methods. In lysis, in most naked viruses, release of virions occur by lysis of the infected cell. Budding is the mechanism of release of enveloped viruses. Enveloped viruses mature by budding. They are not infectious until they have acquired their envelopes. Virus-specified envelope glycoproteins are synthesized by essentially the same mechanisms as cellular glycoproteins. These glycoproteins specified by the virus are inserted into the cellular membranes. Viral nucleocapsids then bud through the membrane at these modified sites and in doing so acquire envelopes. Release of some enveloped viruses occur after budding from the plasma membrane. Virions budding at the nuclear membranes or at any other intracellular membrane are transported across the cytoplasm and then released. Some cellular proteins are responsible for the transport systems. After that, the reinitiation of replicative cycles occur. Usually, viruses released into the extracellular fluid are responsible for initiating new infections. However, infection may also spread by cell-to-cell fusion or particle transmission of the genome to the local cells. So, this is the overall structure of the viral replication cycle. Today's lecture after this, I am coming with another video very shortly.
So, if you like our video, please click at the like button and share this video in your circle. Don't forget to subscribe our channel, which will help you to gather more knowledges in future. And if you already subscribed our channel, then click at our bell button to know our channel updates. At last, please write your experience regarding this video at comment box, which will help us to evaluate ourselves. We are coming with our next video shortly. Till then, goodbye.